Good day everyone, I am Roy C. Borome Jr. Just to inform you, our topic for today is all about history and background of genetics. Now let's proceed to our lesson proper. So history and background of genetics is date back to 1859. So the first discovery is all about the natural selection. In this discovery, Charles Darwin wrote his book about on the origin of species by means of natural selections, or the preservation of favored races in the struggle for life. Following that is the discovery in 1865 with his heredity transmitted in units, in which Gregor Mendel's experiments on peas demonstrate that heredity is transmitted in discrete units. The understanding that genes remain distinct entities even if the characteristics of parents appear to blend in their children explains how natural selection could work and provide support for Darwin's proposal. And after that, the discovery of DNA isolated on 1869, which is Frederick Mischer isolates DNA from cells for the first time and calls it nuclein. After that, in 1879, the discovery of mitosis is described. So in this discovery, Walter Fleming describes chromosome behavior during animal cell division. He stains chromosomes to observe them clearly and describes the whole process of mitosis in 1882. In accordance to the past genetic timeline, in 1900s, the rediscovery of Mendel's work is at spotlight. Botanists De Vries, Correns, and von Chimard independently rediscover Mendel's work while doing their own work on the loss of inheritance. The increased understanding of cells and chromosomes at this time allowed the placement of Mendel's abstract ideas into a physical context, and in 1902, the discovery of chromosome theory of inheritance. Walter Sutton observes that the segregation of chromosomes during meiosis matched the segregation pattern of Mendel's. And also in 1902, the orderly inheritance of disease is also discovered by a British physician, Archibald Garrod, which he observes that the disease alkaptonuria is inherited according to Mendelian rules. This disease involves a recessive mutation and was among the first conditions ascribed to a genetic cause. Later in 1909, the discovery is all about the word gene is coined. So Wilhelm Johansen coins the word gene to describe the Mendelian unit of heredity. He also uses the terms genotype and phenotype to differentiate between the genetic traits of an individual and its outward appearance. Further in the genetic timeline, in 1911, the discovery of chromosomes carry genes. Thomas Hunt Morgan and his students study fruit fly chromosomes. They show that chromosomes carry genes and also discover genetic linkages. In 1941, the discovery of one gene, one enzyme hypothesis. George Beadle and Edward Tatum's experiments on the red bread mold, which is Neurospora crassa, show that genes act by regulating distinct chemical events. They propose that each gene directs the formation of one enzyme. In 1943, the discovery is all about the DNA as a regular periodic structure. William Asbury, a British scientist, obtains the first X-ray diffraction pattern of DNA, which reveals that DNA must have a regular periodic structure. He suggests that nucleotide bases are stuck on top of each other. Further, in 1944, the discovery that DNA transforms cells. Oswald Avery, Colin MacLeod, and Maclean McCarthy show that DNA, not proteins, can transform the properties of cells, thus clarifying the chemical nature of genes. Also in 1944, the discovery of jumping genes. Barbara McClintock, using corn as the model organism, discovers that genes can move around on chromosomes. this shows that the genome is more dynamic than previously thought. These mobile genes units are called transposons and are found in many species. In 1952, the discovery of genes are made of DNA, deoxyribonucleic acid. Alfred Hershey and Martha Chase show that only the DNA of a virus needs to enter a bacterium to infect it, providing strong support for the idea that genes are made of DNA. In 1953, the discovery of DNA, which is a double helix. Francis H. Crick and James D. Watson described the double helix structure of DNA. They received the Nobel Prize for their work in 1962. So, in 1955, the discovery of there are 46 human chromosomes. Joe Hintigio defines 46 as the exact number of chromosomes in human cells. Also in 1955, the discovery that DNA copying enzyme Arthur Kornberg and colleagues isolated DNA polymerase, an enzyme that would later be used for DNA sequencing. In 1958, the discovery of semi-conservative replication of DNA, which Matthew Maselson and Franklin Stahl demonstrate that DNA replicates semi-conservatively. Each strand from the parent DNA molecule ends up paired with a new strand from the daughter generation. In 1959, chromosome abnormalities are identified, which is a discovery in this year. Jerome Lejeune and his colleagues discovered that Down syndrome is caused by trisomy of 21. There are 3 copies rather than 2 of chromosome 21, and this extra chromosomal material interferes with normal development. Also in 1961, messenger RNA ferries information are discovered. Sidney Brenner, Francois Jacob, and Matthew Maselson discovered that messenger RNA takes information from DNA in the nucleus to the protein-making machinery in the cytoplasm. Over 5 years, now 1966, genetic code crack is discovered. Marshall Nirenberg and others figure out that the genetic code that allows nucleic acid with their 4-letter alphabet to determine the order of 20 kinds of amino acids in proteins. In 1968, first restriction enzyme is described. Scientists describe restriction nuclease, enzymes that recognize and cut specific short sequence of DNA. The resulting fragments can be used to analyze DNA, and these enzymes later became an important tool for mapping genomes. Further, in 1972, first recombinant DNA was discovered. Scientists produced recombinant DNA molecules by joining DNA from different species and subsequently inserting the hybrid DNA into a host cell, often a bacterium. In 1973, the first animal gene cloned is discovered. Researchers fused a segment of DNA containing a gene from the African clawed frog, Xenopus, with DNA from the bacterium E. coli and placed the resulting DNA back into an E. coli cell. There, the frog DNA was copied and the gene it contained directed the production of specific frog protein. In 1975, DNA sequencing was discovered by 2 groups, Frederick Singer and colleagues and Alan Maxam and Walter Gilbert. Both developed rapid DNA sequencing methods. The Singer method is most commonly employed in the lab today, with colored dyes used to identify each of the 4 nucleic acids that make up DNA. So in a year, 1976, first genetic engineering company was established. Herbert Boyer founds Genentech. The company produced the first human protein, a bacterium, and by 1982 markets the first recombinant DNA drug, which is human insulin. In 1977, the discovery of introns, Richard Roberts and Phil Sharp's labs show that eukaryotic genes contain many interruptions called introns. These non-coding regions do not directly specify the amino acids that make protein products. So in 1981, the first transgenic mice and fruit flies was discovered. Scientists successfully add stably inherited genes to laboratory animals. The resulting transgenic animals provide a new way to test the functions of genes. Now we're going to 1982. So in this year, GenBank database was formed. Scientists began submitting DNA sequence data to a National Institutes of Health (NIH) database that is open to the public. In 1983, first disease gene mapped. A genetic marker for Huntington's disease is found on chromosome 4. In 1983 also, PCR is invented. The polymerase chain reaction, or PCR, is used to amplify DNA. This method allows researchers to quickly make billions of copies of a specific segment of DNA, enabling them to study it more easily. In 1986, first time a disease gene is positionally cloned. So a method for finding a gene without the knowledge of the protein it encodes is developed. So-called positional cloning can help in understanding inherited disease such as muscular dystrophy. Now let's go to 1987 discovery. It is the first human genetic map. The first comprehensive genetic map is based on variations in DNA sequence that can be observed by digesting DNA with restriction enzymes. Such a map can be used to help locate genes responsible for diseases. Also in 1987, yeast artificial chromosomes was discovered. Scientists discovered that artificial chromosomes made from yeast can reliably carry large fragments of human DNA containing millions of base pair pieces. Earlier methods used plasmids and viruses, which can carry only a few thousand base pair pieces. The ability to deal with much larger pieces of DNA makes mapping the human genome easier. 1989, microsatellites are new genetic markers, are a new discovery. Repetitive DNA sequences called microsatellites are used as genetic landmarks to distinguish between people. Another type of markers, sequence tag sites, are unique stretches of DNA that can be used to make physical maps of human chromosomes. In 1990, the discovery is all about the launch of the Human Genome Project. The Department of Energy and the National Institutes of Health announced a plan for a 15-year project to sequence the human genome. This will eventually result in sequencing all 3.2 billion letters of the human genome. The following year, 1991, the discovery is all about EST's fragments of genes. An express sequence tag EST, an identified piece of a gene, is made by copying a portion of a messenger RNA molecule. As such, ESTs provide a way to focus on the expressed portion of the genome, which is less than 1/10. In 1992, second-generation genetic map of the human genome is discovered. A French team builds a low-resolution microsatellite genetic map of the entire human genome. Each generation of the map helps geneticists more quickly locate disease genes on chromosomes. In 1994, Tomato, or the Food and Drug Administration, approved the sale of the first genetically modified foods. In 1995, the ban on genetic discrimination in the workplace was a spotlight. Protection under the American with Disabilities Act is extended to cover discrimination based on genetic information. Also in 1996, mouse genetic map is completed. The lab mouse is valuable for genetics research because humans and mice share almost all of their genes, and the genes on average are 85% identical. The mouse genetic map increases the utility of mice as animal models for genetic disease in humans. In 1997, the discovery of E. coli genome sequence. The complete sequence of the E. coli genome will help scientists learn even more about this extensively studied bacteria. In 1999, the discovery of chromosome 22 sequence. The first finished full-length sequence of a human chromosome is produced. Chromosome 22 was chosen to be the first because it is relatively small and had a highly detailed map already available. Such a map is necessary for the clone-by-clone sequencing approach. In 2000th century, Human Genome Working Graph is completed. By the end of the spring 2000, HGP researchers sequenced 90% of the human genome with four-fold redundancy. This working graph sequence is estimated to be 99.9% accurate. In 2002, discovery of mouse genome working graph assembled and analyzed. The Mouse Genome Sequencing Consortium publishes an assembled graph and comparative analysis of the mouse genome. This milestone was originally planned for 2003. In 2002, also, rat genome working graph is completed. By fall 2002, researchers sequenced over 90% of the rat genome with over five-fold redundancy. In 2003, the completion of the human genome sequencing. The finished human genome sequence will be at least 99.99% accurate. So the following here are the future and our present today. It will take decades of research for scientists to understand all of the information that is contained within the human genome. In time, more human diseases will be understood at the level of the molecules that are involved, which could dramatically change the practice of medicine by leading to the development of new drugs, as well as the genetic testing to improve and individualize treatments. Now that we have tackled and discussed the earlier history of genetics and its background, why don't we go back to time of prehistory and background of genetics? Now let's start. We have good empirical evidence that humans had an implicit knowledge of genetics from at least 15,000 years BCE. Surprisingly as it may sound, the evidence resides in the remnants of the first attempts at genetic engineering. This engineering took the form of what today is called artificial selection, deliberate breeding of plants and animals for desired characteristics. The best empirical evidence comes from the analysis of pollen in hermetically sealed ancient tombs and from the dog. Further, current phylogenetic analysis suggests that dogs evolved from wolves and were domesticated at least by 15,000 BCE in East Asia. The at least part of these temporal estimates derives from archaeological and DNA evidence of North American dogs, suggesting that they were brought into the continent from Asia by the New World's original discoverers. Artificial selection of the dog for behavioral traits like herding, guarding, hunting, and retrieving is some of the best evidence for genetic influences on mammalian behavior. Like many attempts to manipulate nature, early genetic engineering had unforeseen consequences. Merrill, 1975, reports that the original progenitors for today's onions, lentils, ginger, and many other crops are extinct, probably because our human ancestors selected for fecundity. So-called wild onions and wild gingers are really domestic varieties that got loose and outcompeted their wild-type ancestors. So guys, we just tackled the history and background of genetics. I hope you learned something from our discussion. And you may answer the following questions that will be provided later. Thank you for listening.